Hello, my name is Ksenija Gasic. I'm a peach breeder and geneticist at Clemson University. I would like to thank the organizers for giving me the opportunity to present breeding data management in specialty crops. In this presentation, I will introduce you to Breeding Information Management System, or BIMS, demonstrate its features, and discuss some future works. So, what is BIMS? BIMS is an online tool which allows users to explore the publicly available breeding data in the host community databases, while also providing a data management solution for private breeding programs. Currently, BIMS is available freely in the 4 specialty crop databases: database for Rosaceae, Vaccinium, Citrus, and Pulse, and also in the cotton gen database. This slide features all of these databases. The 4 databases that serve 26 specialty crops had a farm gate value in 2019 of 19.4 billion. They represent 52% of all US specialty crops. BIMS is supported by NRSP10 project and newly submitted proposal to SCRI. And in addition to the database home pages, the databases can be accessed from nrsp.org and breedwithbims.org. This slide lists current functionalities of BIMS, some of which will be demonstrated later in this presentation. Detailed description of BIMS and all its functionalities will be provided in the manuscript that is currently in revision in the database journal. This paper describes how breeders can use BIMS to manage their private breeding data and provides detailed examples. Among the current functionalities are to create the programs, add members, and control permission levels, manage, upload, and edit genotype and phenotype data, to create your own data fields to store and search data, manage the Fieldbook app, perform statistical analysis, perform searches with various categories and trait cutoff values, and archive the entire data at any time. So why BIMS? In addition to described functionalities, there are more advantages to use BIMS. Since BIMS reside in the community databases, curated public breeding data can be imported to your private program. The data includes phenotype and genotype data. Curated genomic data can also be used in your private program. For example, you can search for genotype data using genomic positions of markers without importing those data into your private program. For collaborative projects, all or part of data can be easily released to public when projects end. Another advantage of BIMS is that BIMS is built in Triple. Triple is a toolkit for database construction used by over 100 databases worldwide. In Triple, in addition to core modules, users worldwide can develop and share extension modules. Currently, over 40 extension modules are available. BIMS is developed as an extension module, which means any Triple database can use BIMS. Future analysis tools developed by users worldwide, in addition to work we are doing in NRSP10 project, can be implemented in community databases to be used with BIMS. There are 3 main areas in the BIMS interface. The header region displays the selected crop and program and has links to the configuration page, account page, and the home page of the community database. The second part of the interface is the accordion menu on the left side. This menu allows users to switch between various sections of BIMS. The last region is the tab region, and this is where users primarily interact with BIMS. Each tab in BIMS has an instruction section that provides information on how to use that tab. Without logging in, users can choose a crop and a public BIMS program to view data and explore BIMS. The About BIMS and Help menus provide various information, including links to the BIMS manual and mailing list. To import data into BIMS, the data needs to be in the BIMS templates that are available for download in the template list under the Data Import section of the left-hand accordion menu. Phenotype data can also be imported to BIMS using exported files from Fieldbook. The Data Templates tab lists all the templates with hyperlinks to view and download the template. Users can also download all the templates as a single Excel file. Once the data has been entered into the templates, users can load the data using the Upload Data link. In the Upload Data tab, the file name will appear in the Uploaded Files section after a file has been submitted. To view the progress of an upload job, users can click on the file name and click the View button. The Manage Breeding section lets users view, create, or edit data for trait, location, cross, accession, and trial in addition to program. When users click, for example, trait, the Manage Trait tab opens and shows all the trait descriptors in the trait section. When there is imported public phenotype data, the descriptor groups of both public data and private data are displayed. The admin menu allows changing the order of the traits, adding manually new trait, or merging the same descriptor in different descriptor groups. Clicking one trait opens Trait Details section on the right that shows details of the trait descriptor along with the distribution of the trait values and statistical summary of the full dataset loaded to BIMS. At the bottom of the Trait Details section, delete and edit buttons are available. The Search section allows users to search phenotype, genotype, and cross data. Clicking on Phenotype, the Search by Phenotype tab appears. This tab has three sections: Choose Property, Set Filter, and Phenotype Search Results. The first step to start a search is to select a property or trait under the Choose Property section. For example, users can choose Trial under Properties. After the property is selected, the Set Filter section will populate with options for that data type. After selecting the options, user can click the Add button. The selected filter will then appear in the Phenotype Search Results section. Users can further filter the search results by adding another property or trait under the Choose Property section. Users can further refine the search by adding thresholds for a trait. The Set Filter section will show the data that are relevant for the previous chosen filter. For example, the statistics shown for the fruit L, meaning fruit length trait, is for the accessions from the trials chosen in the previous search. Users can set the minimum and maximum value of this trait, and then click Add, which then adds the new filter to the Phenotype Search Result section. Any selected filters can be removed with the Remove button under each filter. Under the Results section, the Descriptions menu can be expanded to show the filters that were used to generate the initial table. The Data Analysis section of the accordion menu lets users further analyze phenotype data. Here, multiple data sets could be chosen using categories and compare the trait values among the multiple data sets. When users click Statistical Analysis under Data Analysis, the Statistical Analysis tab appears. The tab has three sections: Categories, Traits, and Statistics. Users can select a category under Categories section. In this example, data year, and then choose multiple datasets in the second box that pops up once a category is selected. Categories include the accession list, which is the list of accessions that users saved in the search section. After choosing multiple datasets, users can click the Compare button. When the computation is done, users can select a trait in the middle section, in this example, SSC or sugar content. The statistics section on the right will show a graph with the distribution of the trait values for each data set. A table below the graph will show the statistical values for each data set. Users can click the legend to deselect some of the dataset fields to view subsets. The Fieldbook Management section of the accordion menu provides functionality to generate input files for Fieldbook or load data to BIMS using the Fieldbook output files. Two types of files, the field file and trait file, can be generated using data in BIMS to load to the Fieldbook app. The Configure Fieldbook section allows users to pre-select columns that can be included in the field file. By default, the 3 columns required in the field file, unique identifier, primary order, and secondary order, as well as accessions, are downloaded in the field file. The Fieldbook configuration page allows other columns to be included in the Fieldbook input file so that Fieldbook users can see those in the main screen of the Fieldbook app. The trait link under the Generate Input File section opens a page where it lists all the traits stored in the program. Users can select all or part of the traits to generate the trait file for the Fieldbook app. The field link under the Generate Input File section opens a page with the 3 choices: New Trial, Existing Trial, and Cross. The New Trial section lets users choose one of the accession lists that are generated and saved in the search section, choose additional columns, and generate the field file. Existing Trial section can be used when users want to generate a newly updated field file for the existing trial. The Cross section allows breeders to automatically generate progeny names and generate an input file for a new cross. Breeders can upload a file with the cross name, row, and the number of progeny for each row for BIMS to generate a Fieldbook input file with new progeny names. Customization is one of the very important and unique features of BIMS. In addition to being able to add new traits at any time, BIMS offers various ways to customize the data management. The customization can be done following the links in various pages under the accordion menu. Users can access them through the configuration page link on the top right of each BIMS page. The BIMS Fieldbook link in the configuration page opens a tab where users can change the column names required for the Fieldbook app input file. Users can also add custom columns in templates to store their various metadata unique to their breeding program. Those custom columns can be selected to be used as search categories as well through the search phenotype, cross, genotype category links. The Fieldbook link on the configuration page lets users choose which traits, accession properties, or sample properties will be displayed in the Generate Fieldbook Input File section and resulting Fieldbook input file. Finally, the Trait Order link in the configuration page lets users change the order of traits displayed in the Manage Breeding page. Data in BIMS programs are backed up nightly on-site and off-site for security. The archive section of the accordion menu, however, provides functionality for breeders to archive data at any time. All archived files can be accessed in the user's account page. One of the newest functionalities in BIMS is that it is Brachi compliant, which means you can import data in Fieldbook app to BIMS just by clicking buttons. Some of the future developments are provided here, including management of image data, which is almost done. Next one that we are working on is to make BIMS accessible to breeders without community databases to breed with BIMS.org. Through the NRSP 10 project that is funded, we are working on supporting phenomics data, genome-wide association analysis capability, global performance prediction capability to allow performance prediction based on genotype and environment. Also to develop further analysis tools and breeding decision tools, and to identify sustainability models. In the submitted SCR project, which is pending, we are proposing to further develop Fieldbook app, do more curation of public phenotype and genotype data for the 4 community databases, and on-site training for BIMS Fieldbook app use. This is all I have. We want to thank our sponsors for providing funds and supporting development of this important breeding database. And I hope these presentations have piqued your interest in BIMS and you will check it out. I will be happy to answer any questions. Thank you for your attention.